Hello, and welcome to Reducing Candidate Risk and Achieving Development Success with a Robust Developability Assessment Tool, presented by American Pharmaceutical Review and sponsored by Samsung Biologics. My name is Mike Auerbach. I am the editor-in-chief of American Pharmaceutical Review, and I'll be the moderator during today's event. In this webinar, you'll learn how development platforms help streamline development time and cost, understand the rationale behind using DEVELOPICK in the workflow process, and how DEVELOPICK ensures greater certainty during DSP and formulation development. In addition, this webinar will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note, the right side of your screen also features an overview of this webinar and more information regarding our speakers. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. And now, allow me to introduce today's speakers. Derek Katayama is a lead scientist in the formulation group at Samsung Biologics, where he manages multiple biological molecule formulation projects. Derek has over 15 years of experience in the biopharm industry and has held various positions at companies such as Amylin Pharmaceuticals, MedImmune, Boehringer Ingelheim, and Legacy Biodesign. During his time in the biopharm industry, Derek has worked on a diverse range of projects, including liquid and lyophilized formulation and characterization of monoclonal antibodies, bispecific antibodies, recombinant proteins, protein subunit vaccine candidates, and synthetic peptides. Derek received his doctoral degree in pharmaceutical sciences from the University of Colorado Health Sciences Center, with studies focused on understanding protein stability in both liquid and lyophilized states. Wenchang Lim is the head of formulation development at Samsung Biologics CDO division, where he leads a team dedicated to developing innovative drug formulations. With over 15 years of experience in the biopharm industry, Wenchang specializes in protein drugs and vaccines. He has successfully managed more than 50 formulation development and non-GMP DP manufacturing projects. In September 2022, he helped launch DEVELOPICK, a platform that evaluates target molecules. Prior to joining Samsung Biologics in 2017, Wenchang served as a scientist at LG Chem, where he conducted R&D for purification and formula development in multiple vaccine and protein drug projects. He holds a master's degree in medicine from Seoul National University and a bachelor's degree in molecular biology from Catholic University. Derek and Wenchang, welcome to the webinar.
Thank you. I'm Derek Katayama.
And my name is Wenchang Lim. We both work for the Formulation Development Group in SBL CDO Center.
And so today we're going to share some of our experiences with developability assessment of different molecules. And we look forward to sharing our reasons why we feel that adding a developability assessment at the later stages of discovery can benefit any biopharmaceutical development program. So why invest into late discovery, early development? Well, we all know that there's a pretty high percentage of candidate failure in clinical development. Some aspects to consider that are critical are things such as manufacturability, safety, and stability. Therefore, it is very important to make sure that you select the molecule with the most developable properties by performing a developability assessment early. In other words, by selecting an appropriate molecule for development, this reduces the risk of failure during the development. So at the later stages of discovery, there are often multiple candidates to be evaluated. In order to perform a developability assessment of the multiple candidates, sufficient material is required. And SBL offers a platform called S-CHOSIN, which is capable of producing quality material by transient expression for the purposes of developability assessments. The S-CHOSIN platform not only provides high quality and sufficient material for developability assessments, it also provides an early information regarding productivity using the same host cell as with the stable cell line S-choice. So additionally, the purification of the transiently produced material provides the downstream processing team an initial glimpse into the behavior during purification. So here in this slide, there's a high-level overview of the process for developability assessment, and this is our DEVELOPICK platform. We're at SBL, and as part of our developability assessment, we evaluate molecular stability, relative solubility, and low pH stability. The complete data set allows for the development team to recommend a candidate based upon its physical chemical properties. But let's take a step back here for a moment and consider all of the various degradation pathways for a protein, such as aggregation, particle formation, chemical degradation, fragmentation, and the list goes on. These occur over time at different temperatures, under different conditions, such as shaking, light exposure, and exposure to different impurities. So the sooner we learn about some of the molecule liabilities, the team is better able to select an appropriate molecule to move forward into the development.
Yes, this is a similar strategy to find a stable formulation condition against various degradation pathways. Therefore, the sooner we get information about molecular degradation, the better we will be able to identify a suitable formulation. Even better is if we can select a molecular candidate that does not show as much degradation.
The DEVELOPICK, or the developability platform at Samsung Biologics, is performed at the stage of late discovery. It is at this stage that we can help our clients better understand their candidate molecules before investing into the development. So some advantages of the DEVELOPICK platform are that the analyses are short-term and they use a relatively small amount of material. Therefore, at this stage, DEVELOPICK can evaluate multiple candidates side by side.
That is a good point. Usually at the late discovery stage, material amount can be limited for multiple candidates. Therefore, having a developability platform that is high throughput, that use small amount of material is key to facilitating its implementation.
So what is the developability workflow? So the typical workflow for the DEVELOPICK is sort of as follows, and is shown on the screen. First, we receive the material, whether it's the material provided by the client or it's material that's produced in-house. And the first thing we'll do is we'll do an initial screen or an evaluation of that primary sequence. And then we'll move into looking at a relative solubility assessment. We'll perform some basic characterization and biophysical characterization of the molecule. We'll expose it to some heat stress and some other stresses, such as low pH and high pH.
I think all of these assessments are really important as part of the developability assessment. For example, I recall one project where the molecule was found to be unstable at low pH condition. Therefore, our development team were able to implement a solvent detergent vial inactivation process rather than low pH hold.
Yeah, no, I think that's a great example, Hunshan. I think, and I would note that there's a critical important aspect here, which is communication between groups is seamless. And so, I can foresee this being a tremendous advantage when working with a company like SBL, where the information gained in, say, for example, the late discovery stage can be shared with other functional groups like the downstream processing team. So as I mentioned, the first step for developability is the evaluation of the primary sequence. For the most part, we're looking for hotspots for degradation in the amino acid sequence. So for example, by hotspots, if we see something in the CDR that is labile to degradation, we might make adjustments either in the solution conditions or with excipients to prevent the degradation of that. So for example, if there's an asparagine glycine present in the CDR, this might guide future decisions with regards to the formulation pH. Another aspect of the DEVELOPICK is the biophysical characterization, where we will evaluate multiple candidates oftentimes, and we'll look at different properties for this. And one of the advantages of this biophysical characterization is that it can be done with small volume as well as using automation. So the output from this is we'll obtain information regarding thermal unfolding, both the the temperature of unfolding and temperature of aggregation, the TM and TAG. We'll also obtain hydrodynamic property information, so hydrodynamic size, polydispersity, and so forth.
Measuring these biophysical properties such as TM and irrigation allows us to compare with other molecules like commercial product or molecule in development, some of which we have gone through full development using our process development platform.
Yep. Excellent. So there are some optional biophysical characterizations that can be performed at SBL that are not part of the typical developability assessment. However, we in discussions and communication with the client, if it's desired, we can perform some additional characterization. So for example, we can evaluate the colloidal stability of the molecule by measuring the diffusion interaction parameter, or KD, or the B22, the second virial coefficient. And we can also measure the thermodynamic stability by chemical unfolding, or with using a denaturant like guanidine or urea. In the top panel here, you can see that we have the measured diffusion coefficient as a function of protein concentration. And based on the slopes, you can see that these have different interactions with one another based upon the solution conditions, whether it's pH or the presence of excipients. And on the bottom panel, this is an example of unfolding curve of a complex molecule that we evaluated in the presence of different solution conditions. So kind of following on with some of the additional optional biophysical characterizations, this is just a case study or an example where we measured the diffusion interaction parameter for a series of solution conditions for a monoclonal antibody. And as you can see that we have a variety of, a range of KDs that were measured. And so in this example, we can see that with more repulsive interactions, we saw less aggregation. So it's a way for us to screen different solution conditions to help us identify the most appropriate condition.
Yeah, this is a valuable observation at the early stage of developability assessment, where you observe favorable solution conditions for a molecule with regards to the colloidal stability. And the observation might be able to guide us to understanding which conditions such as pH or buffer type are most appropriate for the molecule.
So a very common stress also applied for the evaluation for different biopharmaceutical candidates is thermal stress. So during the developability assessment, the candidate molecules will be assessed for their thermal stability by incubating the samples at elevated temperature. This will provide a preliminary comparison of degradation rates between the different candidate molecules or of the candidate molecule of interest. It can provide a preliminary assessment of the potential routes of degradation as well, like aggregation or fragmentation. Another aspect of the developability that becomes critical when we consider the final target profile is that of solubility. So at the stage of developability, material amounts are typically fairly limited. So a true evaluation of the solution conditions that favor achieving high concentrations is usually not feasible at this stage. So one methodology that we employ here at SBL provides a measure of relative solubility using a PEG precipitation method. This allows comparing the relative solubility for multiple molecular candidates, as well as evaluating the relative solubility in different solution conditions. So here's an example of evaluating the relative solubility of different molecules. So at the late stage, as I mentioned, there are often several candidates to be evaluated. So by using the PEG solubility assay, we can evaluate all the different molecules and compare them side by side. And it is extremely important to be able to compare the relative solubilities during developability in order to select the most appropriate molecule to move into development.
Some project molecule that we had performed shows low solubility result during the formulation development stages. So we modified formulation conditions in which to improve solubility characterization, and it works. However, if the solubility issue were identified early, it might be possible to choose different molecular candidates that might have better solubility properties.
Yep. And here's an example for relative solubility looking at different solution conditions. So we know that pH or buffer can affect the solubility of a protein molecule. But in this case, the PEG solubility assay was able to compare and determine the relative solubility for a variety of different solution conditions. So in the examples here, we can see that we're comparing different pHs as well as to PBS. And we can see that the lower pH was more favorable for solubility. And so this could be an option with limited amounts of material to evaluate the solubility properties under different solution conditions at the stage of developability. So at the late stage of discovery, there are often multiple candidates for development. Therefore, it's important for us to be able to compare the molecules side by side. And it is also important for us to gain an early assessment of the molecular properties with regards to the physical chemical stability, as well as biophysical properties, solubility, and process stability, like low pH stability that we had mentioned before. Since storage of the formulation conditions can only improve stability incrementally, it is essential that we select the most developable molecule to move forward. It has been a pleasure to be here and discuss with the formulation group leader, Han-Chan Lim, who has been instrumental in establishing and implementing the DEVELOPICK here at SBL. And so it would be wonderful for him to provide the concluding remarks for this webinar.
Thank you, Derek. I want to show the last conclusion for our DEVELOPICK presentation. As you all know, the development of biopharmaceutical is a very challenging task. And there are many aspects of biological molecule that needs to be considered during development. Therefore, being able to select the most appropriate molecule enables success at the start of the project. Samsung Biologics offers both S-CHOSIN and DEVELOPICK for the purpose of helping our clients succeed. Thank you for listening to this presentation.
Yep, thank you very much.
Well, thanks, Derek and Han-Chan, for your excellent presentation. I'd like to remind everyone that you can submit any questions you have for our presenters in the Q&A box on your screen, and we'll get to as many as we can. We have a few questions in, so I'd like to start. Our first question is, so far, what has Samsung Biologics experience with its customers and DEVELOPICK?
Well, the DEVELOPICK platform was launched back in October 2022, and Han-Chan was a big part of that launch. And I anticipate in the future, the DEVELOPICK will be used much more because there's a lot of attention being given to understanding the properties of lead candidates at an early stage of the development.
Excellent. Okay. We have another question here. You mentioned that there is a seamless transfer of information from the developability stage into the process development stage. Can you elaborate a little bit more on this?
Yeah, no, absolutely. That's a good question. So for programs where materials generated for DEVELOPICK at Samsung Biologics, the upstream and the downstream teams are involved early on in the material production, thus giving the USP and the DSP teams early assessment of the molecules. Additionally, as mentioned in the presentation, we also evaluate a low pH stability for the molecule. And this not only helps to inform the formulation team about liabilities for the molecule, but also provides valuable information to the downstream team with regards to viral inactivation, as Han-Chan had mentioned during the presentation, which would employ a low pH hold. And then finally, I think that many of the developability studies that are performed within the formulation development group, so therefore, moving forward, the same SMEs performing the DEVELOPICK on that molecule will probably continue to work on the formulation development. Therefore, any observations of specific degradation pathways like aggregation or fragmentation will be taken into consideration for the selection of the various compositions evaluated during formulation development.
Excellent. Thank you. We have another question. Would you recommend using your transient expression service to conduct DEVELOPICK, and if so, why?
Yes, absolutely. I would recommend using the transient expression system, the S-CHOSIN, for DEVELOPICK service. I think that the material generated has been shown to have good quality attributes as well as mentioned earlier, that the early production of the material by S-CHOSIN can provide early information regarding productivity using the same host cell as the stable cell line, the S-CHOICE.
Mhm. Excellent. Thanks again for your answer. We have another question. The biopharm field is very dynamic and competitive, where speed is often an important factor. Would conducting additional developability studies have an overall impact on the timeline?
You know, that's a good point. I think we are doing more studies, but I think that because the developability assessment can be performed in parallel with other activities, it does not impact the timeline. And the material that we're using for the developability is generated by the S-CHOSIN system, which is also going to be done sort of in parallel. And I think that, you know, to the first part of that, which was with regards to the dynamic and competitive fields, I think that performing a developability assessment is even more important in this competitive and fast-paced world of biopharmaceuticals, because it enables the selection of the correct candidate to move forward into development, and it provides additional information gained from the developability to help all of the processes later in the development stage.
Right. Excellent. Well, I believe that's all of the questions we have for today. We would like to thank Derek and Hung Chang for sharing their knowledge with us, and also offer a special thank you to Samsung Biologics for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand and to share with your colleagues. Thank you for attending the webinar, and enjoy the rest of your day.